Okay, so in this video we're going to talk about the whole overall ideas behind muscle contraction. We'll start with the stimulation of the muscle cell. So initially we have a neuron here, and we can see within our neuron we've got some vesicles floating around, and these contain neurotransmitter. Now this neurotransmitter is really important, but we'll come back to it in a moment. If we just move to this area up here, we can see we've got these calcium channels here. Now this is what's known as a voltage-gated calcium channel. Now voltage gated because when the neuron starts to depolarize, this will cause the channels to open. When those channels open, it will cause calcium ions to flood into the neuron. Now when these calcium channels are flood into the neuron, it causes the vesicles to fuse with the membrane. The vesicles fusing with the membrane will of course release the neurotransmitter into the synaptic cleft. So these neurotransmitters will have been released into the synaptic cleft. So we've got lots of these transmitters. Now they will move by diffusion across. So they will diffuse across the gap. As they diffuse across the gap, they will bind to these channels here. So these channels, these are ligand-gated sodium channels. These ligand-gated channels are normally closed, but once the neurotransmitter binds and sits in this binding site, so we have a binding site here. Once it sits in this binding site, it will cause the channel to open. Once the channel opens, it causes sodium ions to flood in through diffusion. This causes another depolarization. So you have a depolarization of the muscle cell, which is where we now are. So we started off in the neuron, and then we've moved into the muscle cell. Now when this muscle cell depolarizes, we can see we have more channels. Now these channels are voltage gated channels. Now these voltage gated channels will now open. As they open, this will cause calcium ions to diffuse out of the sarcoplasmic reticulum. So I'm just going to put SR, sarcoplasmic reticulum, and they diffuse into the cytoplasm of the muscle cell. Now I'm just going to zoom in over one particular area here. I've done this in quite a bit of detail. And what we can see at the bottom here is we can see an idealized myosin strand, and we have here an idealized actin. So we have myosin and actin, and this little groove here, this groove is a myosin binding site. Now the myosin binding site is important because that's where myosin binds. Once myosin has bound to it, that's when contraction can occur. But under normal circumstances, we have two compounds in the way. We have tropomyosin and we have troponin. So we have tropomyosin and troponin. Troponin has a calcium binding site. So when these calcium ions, which we've previously looked at, when these calcium ions diffuse from the sarcoplasmic reticulum, okay, through one of these channels, as they diffuse through this channel, it'll make its way up to our actin and myosin strands. And what I've added in now is I've shown what's happened. The calcium ion, this red dot here, has changed the shape of the troponin. If we compare its new shape to its old shape, we can see it's changed shape. Now that change of shape has pulled the tropomyosin away from the binding sites. That means the binding sites are now open. So if we just move it along here, I'll just draw another idealized version of this. As we can see, the binding site for the actin is now open. So the myosin head will move into the binding site. And as soon as it's bound to the binding site, it causes a change in the shape of the myosin filament. So what we now have is the myosin filament will be bound. I'll just draw this out again. The myosin filament is now bound into the binding site on the actin molecule. And as soon as this occurs, it causes a change in shape. When that change in shape occurs, like I say, the myosin moves its shape. So it now has the dotted structure. That has essentially moved the myosin filament and shortened the whole fibre. This is known as the power stroke. But unfortunately, from this position, the myosin is unable to dissociate. It's unable to dissociate. So what it requires is ATP to be used at that point to produce ADP and an organic phosphate, and that allows the myosin filament to detach and return to its original shape. You'll now notice that this myosin filament can bind into this binding site and the whole thing will start again, causing a sequential shortening in the muscle fibre. So if we zoom out to just recap the whole structure, the whole thing that's happening. The size of these diagrams are quite important because the actin and myosin filaments are just that small. You can barely see them over there. That's how small they are, and there's thousands upon thousands of them all being affected in the same way. So we start off at the beginning. The action potential arrives, the impulse arrives, that causes calcium ions to move in. We can see here. So this is stage 1. That causes the neurotransmitter, stage 2, to fuse. The neurotransmitter then diffuses across and causes the opening of the channels, which allows sodium in, stage 4. Sodium ions cause a depolarization, stage 5. Opening the voltage gated channels, causing calcium to move out, that would be stage 6. The presence of calcium, although we can't see it on the diagram, causes a change in the shape of troponin. That change in shape causes tropomyosin to be moved away from the binding sites on the actin filament. That would be stage 7. The binding sites, now open, allow myosin to bind, stage 8. Myosin changes shape, the fibre shortens, ATP is required to return it to its original shape, would be stage 9. That summarises the whole of muscle contraction.